Hello everyone, Mr. Linder here. Let's talk about the concept of metabolism, and let's start with the idea of glycolysis and the transition reaction as we look at metabolism of glucose into the production of ATP. So let's start though with the idea of metabolism before we get into glycolysis and the transition reaction. So what is the concept of metabolism when we relate it to the human body? Metabolism essentially is all the chemical reactions that take place in the body. These reactions can extract energy from biomolecules, or these reactions can synthesize or even break down molecules. If we want to further subdivide the idea of metabolism, we talk about catabolic and anabolic reactions. So what are catabolic and anabolic reactions? So catabolic reactions are reactions that release energy through the breakdown of large biomolecules. And so the idea behind catabolism essentially is to take larger carbohydrates or proteins or lipids and break those down into other molecules with the idea that we will release energy from that. Anabolism or anabolic reactions are reactions that utilize energy to synthesize larger biomolecules. So that concept would be producing something like glycogen, or producing triglycerides, or producing proteins, where we're taking smaller molecules and we are building up larger biomolecules, oftentimes for storage purposes or for production of structural components of the human body. So anabolic and catabolic reactions essentially are occurring simultaneously within the cells of our body. We always have catabolic and anabolic reactions taking place. The energy released or stored in chemical bonds is measured in calories. We usually use large C calories when we're talking about human energy. Large calories or large C calories is equivalent to 1 kilocalorie, which in essence is 1,000 lowercase c calories. So it's a unit. It's a unit that we use in order to measure energy within the body. So a kilocalorie or 1 uppercase calorie is equivalent to the amount of energy required to raise the temperature of a liter of water 1 degree Celsius. So when we talk about 1 calorie, we're talking about the amount of energy needed to raise 1 liter of water 1 degree Celsius. So a kilocalorie is 1 calorie, or 1,000 small c calories. During catabolism of biomolecules, much of the energy that gets released is trapped in the bonds of either ATP, or it's trapped in molecules known as NADH and FADH2. So when we talk about catabolic reactions, which is what I'm going to focus on today, looking at glycolysis and the transition reaction, the first two major things that are taking place in what we call aerobic metabolism, we're really talking about catabolic type reactions. And we need to store that energy that's released somehow. So we're going to either store it as ATP, or we're going to store it as NADH or FADH2. NADH and FADH2 are electron carriers. Okay, so these molecules carry electrons. And those electrons can be used later in a process called the electron transport system or electron transport chain. Anabolic reactions transfer energy from temporary carriers to covalent bonds of biomolecules. So the idea of an anabolic reaction is to transfer that energy and use it to build things. Metabolism works through metabolic pathways, so we're going to look at various pathways. This is just a quick summary on this particular diagram, but notice the arrows, right? There's a a pathway, if you will, for converting glucose into phosphoglyceraldehyde. There's a pathway for converting it into pyruvic acid. You'll notice that we can have reverse reactions, so there can be reversible reactions that take place. But there is some sort of metabolic pathway that's going to take place so that we can interconvert molecules. Oftentimes, newly formed products often become the substrates for the next reactions in a pathway. So glucose becomes phosphoglyceraldehyde. That is a new product, but then it becomes the substrate for converting it into pyruvic acid. And then as you look at all these different pathways, you're going to see that there are enzymes involved in the process. And so there are various enzymes within cells that are going to run these metabolic pathways. Some intermediates in a pathway are what we refer to as key intermediates, which means that they will participate typically in more than one pathway. Thus, they will be some sort of branch point to channel a substrate another direction. For example, this phosphoglyceraldehyde is essentially this key intermediate. And you can use phosphoglyceraldehyde in the process of making glycerol, and you can combine glycerol with fatty acids to form triglycerides. So not only is phosphoglyceraldehyde involved in this metabolic pathway, which is known as glycolysis, but it's also involved in a pathway for the production of glycerol and thus the production of triglycerides. So it's a key intermediate. Notice pyruvic acid, a key intermediate. There's interconversion with amino acid pathways. There's pyruvic acid being converted into lactic acid. That's known as our anaerobic pathway. And so pyruvic acid is another key intermediate. Pyruvic acid also becomes acetyl-CoA in what's called the transition reaction. So I want to focus on glycolysis and the transition reaction, look at a couple metabolic pathways, but I want you to understand that there are lots of molecules that can act as key intermediates for other pathways. And so you can branch off and discuss other metabolic pathways when you look at the various substrates that are created through these different catabolic metabolic pathways. So what are some of the catabolic pathways and sort of what is the purpose of looking at catabolic pathways? Well, catabolic pathways are important because they produce energy. So the idea when we look at catabolic pathways is we're actually looking at the idea of ATP production. So on this particular diagram, this is just a summary diagram, but the idea here is that we're looking at a way to produce ATP. If we produce ATP in the presence of oxygen, so if oxygen is available, we would call this aerobic metabolism. Without oxygen, it's referred to as anaerobic metabolism. So we're going to focus on aerobic first. So in aerobic metabolism, we're using oxygen to maximize our production of ATP. If you want to summarize the various catabolic pathways, essentially transferring energy from biomolecules to ATP using oxygen, then we would look at glycolysis. And so this would be the first metabolic pathway. Then we would look at pyruvate becoming acetyl-CoA. This is known as the transition reaction. So again, that's a pathway. After the transition reaction, we would study the citric acid cycle, also known as the Krebs cycle. And then we would look at the electron transport system or electron transport chain. The electron transport chain is actually where we maximize our production of ATP. So for this video series, I want to focus on glycolysis and the transition reaction. So we're going to look at the first two. We'll number these 1 and 2. We're going to look at the first two mechanisms in the process of converting a biomolecule into ATP. What's important to understand is that we focus on glucose when we look at these pathways because glucose runs through all of these processes. Glucose starts at the beginning and runs through all of glycolysis. And then, of course, then we go through the transition reaction, the Krebs cycle, and the electron transport system. But you should note that other molecules can fit into this pathway in various locations. So carbohydrates enter glycolysis typically in the form of glucose, and so that's the starting point. But lipids can fit into these metabolic pathways. Lipids are broken down into glycerol. Lipids are broken down into fatty acids. And so we can transition into these processes at various key intermediate points. For example, fatty acids can be converted into acetyl-CoA. So they can enter into the pathway at this location and then continue on through the Krebs cycle. Glycerol can enter in as phosphoglyceraldehyde. And so it enters in just a little bit below glucose in the glycolysis pathway. Proteins can be broken down into various types of amino acids, and amino acids can fit into various locations of these catabolic pathways. So certain amino acids fit into glycolysis, certain amino acids can become pyruvate, certain amino acids can fit into the Krebs cycle, and so we can generate ATP and we can generate NADHs and FADHs at various locations. So the idea here is that we'll follow glucose through the process, but you can fit in amino acids, you can fit in byproducts of triglycerides, glycerol, fatty acids, and make energy that way as well. As you go through these processes, you'll notice that glycolysis gives us some ATP, the Krebs cycle gives us some ATP, but most of the energy, most of the ATP is going to come from the electron transport system. So before you get to the electron transport system, your goal isn't necessarily to make ATP. Your goal is actually to produce NADHs and FADHs. What the NADHs and FADHs are, they are electron carriers. And so they carry electrons. And they carry electrons to the electron transport system. The goal is to take these electrons to that system so that you can generate a maximum number of ATP. So NADHs and FADHs are going to enter the electron transport system, which is found in the mitochondria of cells in order to generate ATP. Also, as you look at these processes at various points along the way, you're going to see carbon dioxide production. You're also going to have water produced. And so CO2 and water essentially would be considered waste from this particular catabolic pathways. So if we were to summarize aerobic metabolism, it would look something like this. C6H12O6, that is glucose. Okay? Glucose with oxygen will produce carbon dioxide and water. And so these essentially are the waste products of aerobic metabolism. In the process of breaking down glucose in the presence of oxygen, hence aerobic metabolism, we're going to generate ATP. Okay, so the goal here is to generate ATP from these metabolic pathways. So we're going to take glucose in the presence of oxygen, generate ATP, and then generate carbon dioxide and water as quote-unquote the waste products of this catabolic metabolism that's taking place. So I want to look at more detail with glycolysis. So glycolysis is the beginning of the catabolic pathways. And again, glucose is an important biomolecule to use because it goes through every step of these catabolic pathways. The enzymes for glycolysis are located in the cytosol of the cell. So glycolysis takes place outside the mitochondria. So we're in the cytosol of the cell, we're outside the mitochondria, and this is where glycolysis is going to occur. Glycolysis does not require oxygen. Okay, so you can take glucose and create pyruvate or pyruvic acid without oxygen. So glycolysis is a common pathway for both aerobic and anaerobic metabolism. So when you talk about anaerobic metabolism, you essentially are talking about glycolysis as well. Aerobic uses glycolysis, anaerobic uses glycolysis because this is without oxygen. This part right here does not require oxygen. But if you're going to continue through the Krebs cycle and the electron transport system, you need oxygen. Okay? That is, by definition, what aerobic metabolism is. But glycolysis itself does not require oxygen, and so it's common for aerobic and anaerobic. There are enzymes involved throughout the process of glycolysis. And so as you convert one molecule to the next molecule, there's going to be enzymes involved in the process. So let's look at some of the details. So this would be a detailed look at the process of glycolysis. Here we have glucose at the beginning, and then we have our product at the end, which is pyruvate, or also known as pyruvic acid. And you can see that it's a series of reactions. So this is a reaction, this is a reaction, this is a reaction, and so forth. And each reaction has an enzyme that catalyzes from the substrate molecule to the product molecule. So let's zoom in on this and take a look at what's going on. So here we have glucose. And so glucose is our starting molecule that we're going to look at for glycolysis. And the first thing that happens is glucose becomes glucose 6-phosphate. So this is a reaction that occurs. It's in the presence of an enzyme. The enzyme is actually called hexokinase. That would be a good one to memorize. So hexokinase is involved in catalyzing glucose to glucose 6-phosphate. What you should see taking place in this particular reaction is that you are adding a phosphate to the glucose molecule. So here's the phosphate that's been added. Kinases are enzymes that phosphorylate, so they add phosphate to a molecule. And in order to do this, you have to use ATP. Okay, so you are using ATP. We're actually investing energy in the process of glycolysis so that eventually we get energy from the process. So glucose to glucose 6-phosphate, enzymatically catalyzed by hexokinase, and we use up 1 ATP. This also, though, traps glucose within a cell. Right? If we have a cell, and we know that the concentration gradient is glucose is high on the outside of cells and it's low on the inside of cells, how is it that glucose remains low inside of cells? Well, once you turn glucose into glucose 6-phosphate, it's no longer glucose. Okay, this is a new molecule. And so we maintain the concentration gradient of high glucose outside, particularly in our blood, right? High glucose in our blood and low glucose levels in our cells so that glucose can continue to diffuse into the cells. Turning it into glucose 6-phosphate essentially is trapping this molecule within the cell, keeping the concentration gradient moving into the cell so we can continue to get glucose and can continue to make energy. So remember, we used up an ATP though, so we had to invest ATP in this process, but we're going to get that back eventually. The next part of glycolysis is looking at how glucose 6-phosphate becomes fructose 1,6 bisphosphate or biphosphate. There's 2 reactions that take place, so 2 conversion reactions. First, you get fructose 6-phosphate, then you get fructose 1,6 biphosphate or bisphosphate. Notice that now there's 2 phosphates attached to the molecule. So what did we have to do? We had to use up another ATP. So 2 conversion reactions using up another ATP so that we can get to this fructose 1,6 bisphosphate molecule. Once we get to fructose 1,6 bisphosphate, we have used 2 ATP. We haven't generated any energy yet. We've actually used 2 ATP. Once you get to fructose 1,6 bisphosphate, then we can move on to generating what we call phosphoglyceraldehyde. or glyceraldehyde 3-phosphate. Notice that it says number 2 here. That means that we're actually generating 2 of these molecules. Okay, so what's going on here? Well, glucose is a 6-carbon molecule. Okay, glucose is 6 carbon. Phosphoglyceraldehyde, I prefer that name, PGAL, phosphoglyceraldehyde, as opposed to saying glyceraldehyde 3-phosphate. Okay? So phosphoglyceraldehyde is a 3-carbon molecule. So glucose is 6 carbons, phosphoglyceraldehyde is 3 carbons. So in essence, we're able to split that fructose bisphosphate into 2 molecules. And so we get 1 3-carbon phosphoglyceraldehyde, and then we get a second. Okay? So we get a second 3-carbon phosphoglyceraldehyde. And that takes care of the 6 carbons. 3 plus 3 is 6. Okay? So all the carbons are accounted for, but we're able to split, right, this into 2 molecules. So if we're able to split this into 2 molecules, everything from this point on happens twice. So from phosphoglyceraldehyde on, we're actually doing this twice in the process of glycolysis. So 1 glucose, right, is producing 2, right, of these phosphoglyceraldehydes. So let's move on. So in this part of the diagram, what you see here is it says we have how many? We have 2 phosphoglyceraldehydes. And so what you see taking place here, the production of NADH, the production of ATP, another ATP produced, you have to remember that this takes place twice. Okay? So we're actually producing 2 NADHs, and we're producing 4 ATPs, okay? Because this is happening twice. So to make this easier, let's look at a diagram where it actually shows you how it's split. So here we have glucose at the top. Remember, 6-carbon molecule. We made glucose 6-phosphate, we made fructose 6-phosphate, we made fructose 1,6-bisphosphate, and then we created 2 of these phosphoglyceraldehydes. So this is the PGAL molecule. And so now you can watch each molecule go through their metabolic pathway. So for 1 phosphoglyceraldehyde, you produce 1 high-energy NADH and 2 ATPs. And since it's happening twice, here's the other NADH and here's the other 2 ATPs. So what you're looking at for phosphoglyceraldehyde is you're seeing it go through a series of 5 reactions. 1, 2, 3, 4, 5. And those 5 reactions are what produce pyruvic acid. Okay? And because there's 2 phosphoglyceraldehydes, you're going to produce 2 pyruvic acids. So for every 1 glucose, you get 2 pyruvic acids. As phosphoglyceraldehyde goes through its conversions, remember, you are transferring electrons to this NAD molecule. So NAD is becoming NADH, and it's because you're adding high energy electrons to that NAD molecule. And so as you add electrons, the hydrogens are combined with it. Oftentimes you'll see NADH plus H plus written, 2 hydrogens to balance 2 electrons that are added. And so these become high energy electron carriers. And then we also see some ATP production in this series of 5 reactions. The last step of the process is where you make pyruvic acid. So step number 5 here of converting phosphoglyceraldehyde. This is where phosphoenolpyruvic acid, or PEP for short, PEP gets converted into pyruvic acid. So what are the totals for glycolysis? If you're going to look at 1 glucose, and we're going to look at the series of reactions that takes place. So glucose going through a series of reactions, okay, and producing pyruvic acid, what are the totals that we see? Well, we see that we use 2 ATPs, we make 4 ATPs and we make 2 NADHs. And then we also make 2 pyruvic acids, or what we call pyruvates. So if we use 2 and we make 4 ATPs, we actually net 2 ATPs. So glycolysis really gives us 2 ATPs, 2 NADHs, and 2 pyruvates. Okay, so if you're looking at the totals of glycolysis, it's 2 ATPs, 2 NADHs, and 2 pyruvates. Now, where are you going to use the NADHs? You don't use the NADHs until you get to the electron transport system or electron transport chain. So that'll be another video for you guys. What about the pyruvates though? Well, we use the pyruvates in what's called the transition reaction. So I want to show you the transition reaction. What are we going to do with these pyruvate molecules that we just made in glycolysis? And again, you'll only do this transition reaction if you have oxygen readily available. If you don't have oxygen, this pyruvate, okay, would be turned into lactic acid. And I'll go over that in another video. That's known as anaerobic metabolism. But since we're saying we're in the presence of oxygen and we have oxygen available, we can continue with this process of pyruvate going through what's called the transition reaction. So what you see right here is the transition reaction. I'll just abbreviate it TR. Okay? And where is this transition reaction taking place? Well, when you have adequate oxygen, pyruvate will move into the mitochondria of the cell, and the pyruvate will go through this transition reaction in the mitochondria. So this is taking place in the mitochondria of your cell. So pyruvate moves from the cytosol. That's where we made pyruvate, out in the cytosol through glycolysis. It diffuses into the mitochondria, and then we go through this transition reaction. Now, how many transition reactions are going to take place? Well, there's going to be 2 transition reactions because you make 2 pyruvates from glycolysis. And 1 glucose gives you 2 pyruvates. So this reaction here is actually taking place 2 times. It's only shown once, but you have to remember that it's taking place twice. Okay, so 2 pyruvic acids are going to become what we refer to as acetyl-CoA. Okay? We're producing acetyl-CoA, and then it's the acetate unit that's going to move into the next process, which will be the citric acid cycle, or also known as the Krebs cycle. So what's happening in this transition reaction? It's a pretty simple reaction. Let me see. I'm going to show you another picture here. I think it's easier to look at it in the form of a reaction. So this is the pyruvic acid molecule that you made from glycolysis. And what you're doing with that pyruvic acid is you are removing a carbon dioxide. So carbon dioxide comes off as a waste product. And then you are attaching this coenzyme A. Okay, so coenzyme A is being attached to the pyruvic acid as you remove carbon dioxide. And so here we see what's left over from the pyruvic acid. Here's the addition of the coenzyme A, and then here's the carbon dioxide that comes off. In doing so, you are making another NADH molecule. So you're making another high energy electron carrier. The coenzyme A actually is a derivative of a vitamin. It's derived from pantothenic acid. And that coenzyme A is actually used over and over and over again within the mitochondrial matrix. So if we go back to this diagram, this represents that idea that you can recycle, right, that coenzyme A. So here's pyruvate. We're going to add coenzyme A to pyruvate. Carbon dioxide is coming off as a waste product. We're producing NADH, a high energy molecule because it gains electrons. And in the process, you're making acetyl-CoA. The coenzyme A will break off as the acetate unit is moving into the citric acid cycle. So this happens twice. This transition reaction happens twice. And what is the purpose of the transition reaction? Well, it's essentially to turn pyruvate into acetyl-CoA, and we get two of them because we have two pyruvates, we get two acetyl-CoAs. And since we make one NADH per pyruvate, then we make two more NADH molecules. And then we get two carbon dioxides as waste. Okay? And we know that humans exhale carbon dioxide. So we produce carbon dioxide in our cells. It's dumped into our circulatory system. We take it to our lungs, and then we remove carbon dioxide from our body. So we bring in oxygen to run aerobic metabolism, and we exhale carbon dioxide as a waste product. So the summary of the transition reaction is to get two acetyl-CoAs, two NADHs, and two carbon dioxides as waste. And so if we're adding up what we've done so far, is we've been able, through glycolysis and through the transition reaction, we've been able to produce 2 total ATP and 4 NADH molecules. Right? We got 2 NADHs from glycolysis, right? 2 of the NADHs came from glycolysis, and 2 NADHs came from the transition reaction. So although we've only made a couple ATP, we've made 4 of these high energy electron carriers that we are going to move towards the electron transport chain, and we're going to use those to maximize our production of ATP. And so we'll talk about the Krebs cycle or citric acid cycle and the electron transport chain in another video. But I hope this helps with glycolysis and the transition reaction. Take care.